All right, so chapter two, the main thing we're talking about here is the micro aspects of the nervous system, the small aspects of the nervous system. At the end of this chapter, we will be talking a little bit about some of the macro, bigger aspects of the nervous system. But kind of the idea there at the end of the chapter is that some of the brain regions that we're going to be studying in more detail later on in the semester, we're just going to have a brief exposure to some of those brain regions and their putative functions now before we get into learning about some of the details of these various aspects of the brain in future chapters. So last time we were talking about one aspect of the micro aspect of the nervous system, which is neurons and the way they communicate using action potentials. Just to kind of briefly go through some of these terms. Membrane potential is the charge inside the membrane. You can think of this as the cell body of a neuron at any given point in time. The resting potential is a special membrane potential, usually around -70 millivolts, which is when the neuron is not receiving any excitatory inhibitory neurotransmitters. There's a tendency for the charge, the membrane potential, to be around -70 millivolts. You can see that right here. That's -70 millivolts. The threshold. Can anyone tell me, just to see if you guys remember, what happens when that threshold of -55 is reached? Anyone remember what happens inside the neuron when that threshold of -55 is met? Anyone remember this?
Isn't that when an actual action potential will start to propagate down the axon? It's like that has to be met in order for it to occur.
Exactly. Yeah, well said. Thank you. Exactly. When that threshold, you can see it right here, that threshold of -55 is met. This causes the action potential. The action potential is represented in this diagram by a quick change in the voltage that goes from negative, that -55 threshold, quickly to the positive side before it goes back down to the resting potential again, which is at -70. So you receive enough of an excitatory neurotransmitter, and that will cause the membrane potential to reach this threshold, which will then cause a quick change in the voltage towards the positive side in the neuron. That change in the voltage lasts for about 2 milliseconds, so a fraction, a sliver of a second before things get back to the resting potential. That is the action potential, this change from negative to positive and back to the resting potential voltage. Those two neurotransmitters talked about last time. Glutamate is the neurotransmitter that will make the voltage inside the neuron more positive. Glutamate can make the voltage closer and closer and closer to this threshold. And then once the voltage reaches that threshold, the action potential fires. GABA, on the other hand, an inhibitory neurotransmitter, will make the charge inside the cell even more negative and more negative, which means that the cell is getting further away from an action potential. So glutamate increases the likelihood that the neuron fires an action potential. It is excitatory, whereas GABA decreases the likelihood that it's going to fire an action potential. It is an inhibitory neurotransmitter. So here is the term receptor. An AMPA receptor is in particular a type of neurotransmitter that responds to glutamate. And an AMPA receptor is it works by quickly changing the polarity, that is the membrane potential, in the postsynaptic neuron. So anyone want to take a guess? Let me give you a multiple choice question. Given what I've talked about, AMPA receptor, let's see, right here. Which part of the part of the neuron that I said is the receiver? It receives messages from other neurons. It's important to kind of understand or to think about which part of the neuron is going to have these AMPA receptors. The AMPA receptors are basically on the part of the neuron which is holding out its hand waiting for a message. And those AMPA receptors are found on the dendrites right here. AMPA receptors receive neurotransmitter, in particular glutamate, and they will change the polarity, they will change the membrane potential inside the cell. Oops. So I bring up these AMPA receptors now because I want you to understand their basic function. What an AMPA receptor does is it changes the membrane potential of the postsynaptic neuron, the neuron that is receiving the message. When it receives a neurotransmitter into the AMPA receptor, which is a certain, you can think of it as a specific type of lock that occurs, or a specific type of receiver on the dendrite. When a neurotransmitter activates that AMPA receptor, what it does is it will cause a change in the membrane potential, the polarity, voltage inside that neuron. And I'm going to contrast that with a different type of receptor that we're going to talk about in a second. So it's a little bit of foreshadowing. Before that, I just want to talk a little bit in more detail about kind of the dynamics of action potentials, which is definitely we're now going into definitely more detail than you probably would have if you learned about action potentials in like an introductory psychology class. So here are two terms which the book talks about. Gradient basically just means difference. Concentration gradient means a difference in the chemicals on the inside versus the outside of the neuron. And I'll show you what two chemicals, and then those two chemicals we're interested in are potassium and sodium. And there's a difference such that one of those chemicals is more common, has more of it on the outside of the neuron, and another one of those chemicals is more common inside the neuron. So this is a concentration difference, a difference in the amount of chemicals inside versus outside the neuron, versus an electrical gradient, which is a difference in charge. We've already talked about this. The resting potential is an example of an electrical gradient. The resting potential is a negative 70 millivolts inside the cell relative to the outside of the cell. So what this means is the charges are different inside the cell versus outside the cell. It's more negative inside the cell compared to out. And it's kind of important to now start thinking about the idea that the fact that the resting state of a neuron is negative is kind of interesting, because in physics, there's a tendency for systems to reach equilibrium. And what equilibrium is when the charge in both sides of the system, inside and out, is zero. That's kind of equilibrium. That's when in nature things are at rest, when there is no gradients at all. But in a biological system, or at least in neurons, the resting state is actually a gradient, which kind of, I don't want to say it defies the laws of physics, but it's interesting. Something has to be going on there, because left alone, physical systems will tend to reach equilibrium where there's a charge of zero. But the resting state, kind of like the baseline state of neurons, is an electrical gradient, a difference of charge of around negative 70. That is the resting potential. And so we need to start thinking about how is this accomplished? How is the resting state of a neuron, the resting potential, how is this not zero? What is the mechanism by which this negative voltage is maintained? The answer to that is this thing called a sodium-potassium pump, which is shown right here. The sodium-potassium pump is a thing inside the membrane, basically the cell wall of neurons. And what it can do is it uses ATP. And ATP is basically, it's a term, if you take an introductory biology course, you'll learn a lot about ATP. For this course, it's sufficient to think of ATP as basically just energy. When you eat food, the sugar and the nutrient, we'll say the sugar in the food basically turns into ATP, which allows your body to do work. It is energy that the body can use. And ATP is used by the sodium-potassium pump. And what it does is for every unit of ATP, it sends 3 sodium ions. And please notice those are sodium. Those sodium ions are positively charged. Each one has a charge of +1. So it sends a charge of 3 positives outside the cell, while it simultaneously brings in 2 potassium ions into the cell. So it uses energy. You eat food, that food is converted to energy, ATP, and then your body uses ATP to kind of go against the tendency of a physical system to go into equilibrium. Usually, once again, equilibrium is a zero voltage. But I've already said the resting state of a neuron is a negative voltage, an electrical gradient. The way that is accomplished is that your body uses energy, it uses ATP, and for every 3 positively charged ions that are sent outside the cell, making the outside of the cell relatively positively charged, only 2 positively charged ions of potassium enter the cell. So for each unit of ATP which is used, the charge inside the cell becomes relatively more negative by a unit of by 1 compared to the outside of the cell, because 3 positive charges leave the cell, only 2 positive charges enter the cell. This also causes a concentration gradient such that the chemical, the sodium ion, is more common outside the cell, whereas the potassium ion is more common inside the cell. So this is important. This is probably a new thing that you wouldn't have learned about in an introductory psychology class. The sodium pump uses ATP to make the resting potential -70, to make it negative. Without the sodium potassium pump, presumably the charge inside the cell would be 0. It'd be at equilibrium. But your body uses energy so that the resting potential inside the cell is negative. And so now I'll focus on ion channels, which are shown over here. Ion channels are the thing which are responsible for the action potential. There's kind of a series of opening and closing of these ion channels that occur on the membrane of a neuron, basically the wall of the neuron, which can cause ions that are positively charged, these sodium potassium ions, to either enter the cell or leave the cell. So if you think about this, we have all these positively charged ions outside the cell, and it's negatively charged inside the cell. And you can, if a certain type of door opens one of these ion channels, what it's going to cause, because of the electrical gradient, is going to cause all those positively charged ions to enter the cell. The reason that happens is in the laws of physics. If it's really positive out there and it's really negative in here, those positive ions want to get to the area where it's negatively charged to reach equilibrium. And so ion channels, they rely on the electrical force. When the channel opens up, those positively charged ions outside the cell will come into the cell, which will help the cell get equilibrium. And that's, going back to this, that's how this going to positive occurs. Equilibrium is right there where 0 charge. When you get to this -55 threshold right here, what happens is a specific type of channel on the membrane, which allows sodium to move through, that channel opens up, which causes an influx of positively charged sodium ions to enter the cell, which causes a shift, a quick shift in the positive direction, and it actually overshoots. That's why they call it overshoot. It actually overshoots equilibrium, and it goes to somewhere on the positive side here. In this diagram, it goes to +40. And then at that point, the sodium channels close, and the neuron will then go back down to the resting potential, which is -70. So that's not the whole story, though. So I've just talked, let's see. Yeah, when the -55 threshold is reached, sodium channels open up, which causes all this sodium, Na plus, outside the cell to enter the cell, which causes the cell to become temporarily positive. But then kind of at the peak of the action potential, when the voltage inside the cell, inside the neuron, is most positive, then a different type of ion channel opens up. The ion channel that opens up is the voltage-gated potassium channel. Please note that at the peak of an action potential, the charge inside the cell is actually very positive. And inside the cell, there's also a lot of potassium, because that's what the sodium potassium pump does. It makes a lot of potassium move inside the cell. So at the peak of the action potential, the inside of the neuron is very positive, and there's also a lot of potassium. At the peak of the action potential, those potassium channels open up, and that causes a lot of the potassium to leave the neuron. And that's the way that the neuron gets quickly back to the negative side of things. So it gets super positive during the action potential when the sodium channels open up, causing a bunch of sodium to rush in, moves it way to the positive side. Once it gets way to the positive side, then the potassium channels open up, which causes a bunch of potassium to leave, which causes the charge inside the cell to come back down towards the negative side. And all this time, the sodium-potassium pump is doing its thing by keeping the resting potential negative. It moves 3 sodium ions outside the cell for every 2 potassium ions that enter the cell. Here's a diagram that's in your, I believe this one's in your textbook, but if it's not, I think it is a really good diagram for kind of visualizing the series of events that occurs regarding these ion channels during an action potential. So the red trace here is the action potential. The red trace is just measuring the voltage inside the neuron at any given point in time. The resting potential is negative 70. The threshold of negative 55 is reached, which causes a quick burst to the positive side, and then things go down. This black, the solid black trace right here, this is the sodium channels. Ionic conductance basically means how much of a given ion is moving across the membrane. At the beginning, and especially at the peak of the action potential, the sodium channels are open, but then right around the peak, then the sodium channels close, and you can see that here, the sodium channels are closing, and as the sodium channels close, the potassium channels are opening up. Any questions or comments about sodium potassium channels and the sodium potassium pump? Okay. So I don't have I've kind of already said this stuff about neurotransmitters. This diagram right here is also taken from the book. It's just showing, yeah, I do like this. It's showing an electrode in the postsynaptic neuron, measuring the voltage, and it shows the resting membrane potential is negative 70. Neurotransmitters from the presynaptic neuron, so that's the sending neuron, those neurotransmitters travel across this gap. By the way, can someone remind me, what is the name of the gap between the presynaptic neuron and the postsynaptic neuron? That's a definitional. Can anyone remind me what this gap is right here, what it's called? This space between yeah, go ahead, please.
The synapse.
That is correct. Presynaptic means before the synapse. The synapse is this space right here. Postsynaptic means after the synapse. So the presynaptic neuron sends neurotransmitters across the synapse, and then the postsynaptic neuron receives those neurotransmitters. These things right here are the neurotransmitter receptors. I've already talked about one, the AMPA receptor, which especially responds to glutamate, an excitatory neurotransmitter. What the AMPA receptor will do is it will change the polarity inside the postsynaptic neuron. And you can see an example of that here. The resting potential of this neuron is negative 70, as I've mentioned before, but then neurotransmitter travels across the synapse. You can see that down here, where it's received by these receptors. It doesn't actually specify what type of receptors these are. But then you can then see that the charge inside the cell changes. It becomes here. We can tell, well, this is something to think about. Is the neurotransmitter, based on what we see here, is this neurotransmitter an excitatory or inhibitory neurotransmitter? Can anyone tell? Is this neurotransmitter that we see here, based on what it's doing right here, can we say that that neurotransmitter is an excitatory or inhibitory? Oh, shoot, it says it right there. Darn it. It's an excitatory neurotransmitter. You can see it right there. But the question is, seeing here, it says it's excitatory neurotransmitter. How do we know? What can we see here in this graph that is indicating that it is an excitatory neurotransmitter? Can someone tell me what's going on there that we can see that this is an excitatory neurotransmitter?
It's making them
Oh, I heard two people. I don't know how to select. One person just talk.
It's making the charge more positive as opposed to negative.
Exactly. An excitatory, it starts negative, but then when it receives a neurotransmitter, there's this bump, and which is moving it towards the positive side. It's moving it more positive. An inhibitory neurotransmitter, then on the other hand, would move things down. So we know this is excitatory because it's moving the charge up in the positive direction. But we know that this has a caused an action potential yet, because if it caused an action potential, then the charge would temporarily go way to the positive side before it comes back down. So this is an excitatory neurotransmitter that, at least in this diagram, has not yet caused an action potential, because if we had an action potential, it caused a big boost in positivity for a couple of milliseconds, which is not present yet in that graph right there. Questions or comments then about excitatory versus inhibitory neurotransmitters?
So can it be excitatory but not have an action potential go? And so there wouldn't really be like if you wanted to like move a muscle but it didn't make it, like you didn't end up moving it, could it possibly be excitatory but just not enough to make it an action potential, so then the muscle wouldn't really move?
Definitely. That is, as far as I understand, very, very true. That neurotransmitter works by kind of accumulating, and you need to have enough of an excitatory neurotransmitter to cause an action potential. And if you don't have enough of a neurotransmitter, or if you don't have enough receptors to receive that neurotransmitter, then it might not have enough of a change in the positivity of the charge inside the cell to cause an action potential. So the way to think about it is you have to have enough, you have to have a lot of an excitatory neurotransmitter before that will cause an action potential. Good question. Anything else?
Does it have to pass 0 for it to be an action potential, or does it not have to pass?
I believe by definition it passes 0. I believe action potentials by definition have an overshoot, where it becomes temporarily more positive. It becomes on the other side of 0, on the positive side of 0. There could be exceptions though. There's many neurons in the nervous system, and not all neurons fire action potentials in exactly the same way. There's a tiny bit of variability. But my understanding is that most neurons will overshoot. They will go beyond 0 temporarily before they return back down to negative. Okay, so this is the reason long-term potentiation is the reason I was talking about AMPA receptors earlier, is there's another type of receptor, NMDA receptor. And these two receptors, they're both on the postsynaptic neuron. So they're both on the receiving neuron, but they do different things to the receiving neuron. The AMPA receptor, this is the one that does what we were just talking about. You receive neurotransmitter, it can either have an excitatory effect on the neurotransmitter, that is make the charge inside that neuron positive, or if you receive an inhibitory neurotransmitter, it can make the charge inside the neuron relatively more negative. And so that's like the AMPA receptor is an example of a type of receptor on the postsynaptic neuron. And all it does is it does it quickly, is it will change the voltage inside the postsynaptic neuron. But this quickly thing, this is a term which is included here on purpose. It very quickly, in a matter of like a fraction of a second, I receive glutamate in an AMPA receptor almost instantaneously or very quickly, in a matter of milliseconds. It's going to change the voltage in the neuron that received that glutamate into the AMPA receptor. Very quick changes in the polarity inside the neuron when glutamate is received by the AMPA receptor. What AMPA receptor does is it changes the polarity in the cell. AMPA is an example of one that receives glutamate, so AMPA receptors tend to have an excitatory effect on the postsynaptic neuron. Contrasting that with an NMDA receptor, it uses glutamate, so it uses the same neurotransmitter, but it causes different types of changes in the cell. NMDA receptors, if I'm an NMDA receptor and I receive a glutamate, it doesn't just cause the neuron that received that glutamate, it doesn't just make it more positive, the charge inside the neuron more positive. NMDA receptors, they have a more slow effect on the neuron. This video, if you click on this video, it's a 2-minute neuroscience video. It actually does a pretty good job explaining this. I'm just going to talk about it briefly, and I do encourage you to watch this video. It actually goes slightly more detailed than I would require you to know as far as the molecular mechanisms which support long-term potentiation, but it does a good job of explaining, especially the second half of the video, the second minute, does a good job of kind of explaining what NMDA receptors do for the neuron. And I also say here, though, one of the the two things the NMDA receptors do is that if I'm an NMDA receptor and I receive glutamate, that can send messages to the cell and to the genetics of the cell, the genes which are inside the cell body, to produce more AMPA receptors. And so this is not a quick thing. The body doesn't just produce AMPA receptors in a fraction of a second. Glutamate is received by these NMDA receptors, and then it can cause a series of these chemical and genetic interactions, which we definitely don't have to understand for this class, which can cause then changes in the way genes inside that neuron are expressed. And one of the most common types of changes that can occur is that it can cause the cell to produce more AMPA receptors. And this is what this video does a really good job of explaining. If you have more AMPA receptors, basically what that does is that makes that neuron more sensitive to glutamate when glutamate is received sometime in the future. Someone earlier just asked, do you have to receive a lot of a neurotransmitter before you can fire an action potential? And I said, yes. But this is kind of another aspect of this. If I have a lot of AMPA receptors on the postsynaptic neuron, then that makes it so that the glutamate has a more of an excitatory effect on the neuron which is receiving that. If I only have one AMPA receptor, it's going to take a long time for me to receive enough glutamate where I'm going to fire an action potential. But if I have hundreds of thousands of AMP receptors on my neuron, then I'll be able to fire action potentials much more quickly. So what NMDA receptors do is they receive glutamate, they can cause more AMPA receptors to be constructed, which in the future then can increase the likelihood that that neuron will fire an action potential when it receives enough glutamate. So there's kind of an interaction between these two. AMPA receptors influence the likelihood of firing action potential very quickly. NMDA receptors influence the likelihood that AMPA receptors are created, are constructed by the neuron. So watch this video. It explains it slightly differently than I do, and it uses illustrations, so that will help you, I think, studying for the test and stuff and just understanding this. Any questions, comments about AMPA versus NMDA receptors?
Is there a way that the NMDA receptors, like the AMPA receptors that it creates, for those to be like turned off, or once those are created, are they just always there and always present?
Which one?
The created AMPA receptors.
So what is your question about the AMPA receptors again?
Like once the NMDA receptor creates more AMPA receptors, are those always active?
I think so. I just don't know the answer to that, though. I'm afraid I'm going to have to say I don't know for that one. I would guess that yes, they are. Though psychopharmacologically, there are probably drugs which could be introduced into your nervous system, which could have effects on the functioning of glutamate. That would be a hypothesis, at least. My understanding is that the AMPA receptors are typically on, but I imagine there are probably pharmacological interventions that could temporarily change that. Okay, so another topic, electrical synapses. These are interesting. At least to me, I've always found these to be very interesting. There's been research on them in the last 20 years, but they're still kind of a mysterious aspect of the nervous system. So most synapses in the nervous system allow chemicals to cross. So, we talked about the chemicals that cross the synapse of most neurons are chemicals, neurotransmitters. But there's a special type of synapse where it's not chemicals that pass across the synapse. Instead, it is an electrical signal. So we've talked about for action potentials, usually there's kind of like the two aspects of the communication. There's the electrical propagation down the axon, which then eventually is received by the axon terminals. That's the electrical aspect of communication in an action potential. But then the axon terminals release the neurotransmitter chemicals, which are then received by the postsynaptic neuron, which potentially can cause the electrical signal to increase again. The action potential, the electrical aspect of the communication can occur again. So in normal neuronal communication, there's a chemical component of communication, and there's also the electrical component of communication. But for electrical synapses, there is only the electrical component. There is no chemical component at all for electrical synapses. And you can see the electrical synapses right here. The book calls these gap junctions. So gap junction is actually in biology, it's a more general term. I don't really understand the gap junctions outside of the nervous system, but my understanding is that gap junction is basically a word for a type of molecule that can have functions all over the body. But in particular, gap junctions in the nervous system are electrical synapses. And one of them, this whole thing is a gap junction, but you can see these pores right here. This whole thing is an electrical synapse, but you can see these pores right here. And what these pores are doing, they're basically openings between one neuron's cell body, which is out here, or what is, I'll just say one neuron, which is out here, and another neuron, which is down here. So we have two neurons, and they're the inside of their cell bodies, the inside of those two neurons is basically directly connected to the other. You can kind of think of them as the two neurons are like plugged directly into each other. And the connection occurs via these pores, which are literally just openings. But what these pores allow is for electrical communication to occur directly between these two neurons. Neurotransmitters are not passing through these gaps right here. Instead, it's electrical activity is directly shared between cell bodies of two neurons. And so the reason why this matters is because well, at least the reason why in theory these things matter is because in the nervous system, in the brain in particular, a lot of the cognitive things that occur, a lot of our thoughts and a lot of our behaviors are influenced by populations of neurons working together at the same time. And so population of neurons means there's a whole lot of neurons, hundreds of thousands or maybe even millions of neurons that are working together to help you represent a thought or to represent a plan or to represent some type of behavior. And what electrical synapses appear to do is allow the synchronizing of the electrical activity, the synchronizing of the action potentials and the electrical fluctuations of populations of neurons. So synchronizing, this actually seems to be the main thing that electrical synapses, that is gap junctions, do. They allow populations of neurons, for all the neurons, the electrical fluctuations will occur in synchrony at the same time, which can cause all of those neurons to fire action potentials at the same time and go back to the resting potential at the same time. So it's kind of a way of organizing the behavior of all of these neurons in time, making them kind of like doing a dance routine where they're all doing the moves at the exact same time. But yeah, and another thing about action potentials, which is an important fact, is that they have their influence quickly. So with a normal chemical synapse, it takes a while for the neurotransmitter to diffuse across the synapse. It takes not, tons of time, it's still less than a second for a neurotransmitter to cross the synapse, most likely. But the amount of time it takes neurotransmitter to cross a chemical synapse is much greater the amount of time that electrical activity will cross a gap junction. For all practical purposes, the changes in electrical activity across a gap junction are almost instantaneous, like a fraction of a fraction of a fraction of a millisecond. If the voltage change over here in this neuron, then almost instantaneously there will be a change in the voltage of this neuron. This is in contrast with chemical synapses, where I have neurotransmitter which is released and has to somewhat slowly diffuse across the synapse before it is received by the postsynaptic neuron. So electrical synapses, that is gap junctions, important for synchronizing and for speeding up neuronal activity. I'm going to present some research that in a little bit more detail about this thing called the connexin 36 protein, which has been, as far as I've been able to observe, the primary way that researchers have been able to study these gap junctions, these electrical synapses, and their function in the nervous system. So here's an abstract. So if you can read this abstract, I'm not going to read it to you now, but this comes from an actual article in Annual Review of Neuroscience, which is a pretty darn good journal. It's one of the top rated neuroscience journals, that there is. And it's a review article about electrical synapses. In particular, they're talking about this protein, which they call connexin 36 or CX36. And basically, according to this idea, in order for the gap junctions to work, in order for the electrical synapses to work, this protein, CX36, is doing something. It's not exactly clear the mechanism by which this protein is helpful to the electrical synapse. That is not exactly clear. But what is clear is that CX36 is doing something for the electrical synapses. In particular, CX36 is important for the electrical activity of different neurons, which are connected via electrical synapses, to synchronize. And I'll show you that right here. WT, I believe these are mice. This basically represents right here, these two waves are, the top one is one neuron and the bottom one is a different neuron. And these waves are fluctuations in electrical activity. WT stands for wild type. You can think of this as like a typical, intact, normal animal, which, by nature, normally we have CX36, in our bodies that allow, that's the normal state of animals. We have CX36 that allows our gap junctions to work. Contrasting that with CX36KO. KO means knockout, which is a term which is presented, I believe, in chapter it's either in chapter 1 or 2 or it's one of the first few chapters. This is a term, knockout, which is talked about in one of the early chapters of the textbook. But a knockout is basically, you can use a genetic manipulation which will make it so that CX36 is not expressed. So CX36 is the amount of that in the body is reduced. And what the researchers wanted to investigate here is in an animal which has CX36, which therefore is able to have functioning electrical synapses, is the synchronization of neurons different than an animal whose CX36 is knocked out, whose electrical synapses are not working. So once again, the black trace is one neuron, the gray trace is a different neuron. And what we're looking for is, are the electrical activities of these neurons synchronized or not? And here you can see that in the wild type neuron that has CX36, they're synchronized. And you can see that because they're like perfectly interlocking. When the charge of one neuron increases, the charge of the other neuron, which is coupled to it by a gap junction, increases as well. And then when one neuron decreases, the other one decreases as well. So the charge of these two neurons, which are connected via an electrical synapse, that is a gap junction, they're fluctuating exactly. They're like, once again, they're kind of like dancing together. When the electrical charge goes this way in one neuron, the other one does the same thing, and they're just moving in step. They're moving in synchrony. That's the normal state of things when electrical synapses are working. And the CX36 knockout, that is when that protein is removed or is in very, very low concentrations in the body, you can see that these same two neurons are relatively less synchronized because they're not perfectly interlocking here. Here, for instance, this neuron's going up while there's certain neurons going down. They're no longer synchronized here. And there's actually a lot of research now which supports this idea that one of the main functions of electrical synapses is to cause the electrical signals in neurons to synchronize very, very closely in time. Questions or comments about this study and electrical synapses in general? Okay, so that's it for micro stuff, I believe. Yeah. So now for the rest of this chapter, we're just going to be talking about some of the big picture macro things again. And this is going to be the dominant theme of the book. The micro stuff is interesting, but that's more on the behavioral neuroscience side of things. In a cognitive neuroscience class, usually you're talking about bigger picture, the macro side of things, mainly because if you're trying to connect cognitive function to the brain, it's been easier for researchers to connect cognitive function to the macro aspects of the brain rather than to the micro, like an individual neuron. That is, thoughts and plans and behaviors seem to be represented more on the collective behavior of many neurons on the macro side of things, rather than in the behavior of an individual neuron. When you think, it's not really just a single neuron doing that thinking. It's a whole bunch of neurons that are doing the thinking collectively. They work together to produce a thought or a plan or an action. So we'll be talking more about the macro side of things. And so just some basic terms really, so to help us understand That will hear the divisions of the nervous system, the different aspects, the different types of signals that can be sent throughout the body and the brain. So you probably heard most of these terms before, but I'll review them anyway. Central nervous system is comprised of the brain, which is obviously up in the head, and the spinal cord right here. The peripheral nervous system, on the other hand, is the neurons, which is basically a synonym for nerves in the body that are not the brain and are not the spine. And so it's a little bit hard. There's not tons and tons of pictures of the anatomy of these peripheral neurons, but this is one decent one. And so all these little wire looking things here that are in the body. So you have some going into the arm, you have some going into the leg, in this arm. Those are peripheral nerves. I also want to point out there are definitely nerves which go into the core, the trunk of your body. For instance, I know specifically the vagus nerve is a nerve which goes from the brain to the heart. And there are also nerves which go towards your lungs. And there are definitely nerves which travel. You can't really see them here, but to your gut. There's definitely connections between the brain and the gut. So there presumably are nerves that you can't see that are emanating from the spine, which are traveling in this direction, around like where your intestines and your stomach would be up here. So there's nerves kind of innervating all of the different organs of your body. So the nervous system isn't just for thinking and moving your arms and stuff like that. It also controls kind of the functions of your internal organs. And this is what the autonomic nervous system, this is the name for those aspects of the nervous system that regulate the activity of organs and general physiological processes. So this is something that in my 625, a Psych 625 class, that will definitely I teach in more detail. For instance, we'll study in that class how the behavior of the heart changes as a function of your emotional state. And so a very simple idea here is, like, for instance, when you're fearful, your heartbeat will tend to increase its rate of beating. So this would be an example of the sympathetic nervous system. So the autonomic nervous system is just those aspects of the nervous system which regulate the activity of organs like your heart or your stomach, your intestine. The sympathetic nervous system is that aspect of the autonomic nervous system, which is highly activated like when you're stressed. So when you're stressed, your heart rate will tend to increase. And when you're stressed, things like digestion also will slow down. When you're stressed, your body is in more of a state of trying to stay alive. And at that point, the energy that your body was spending on digesting food, digesting food doesn't seem quite as important when you're in a situation of stress. And so your body, if you're stressed, if you're worried about getting eaten by a predator, your body will take the energy that it was using previously to digest food, and then it will make it so that energy can be used to help you avoid getting eaten or injured otherwise. And so that is what the sympathetic nervous system does. It changes the goals of your nervous system towards fight or flight. I said flight or flight, but it's supposed to be fight or flight right here, dealing with the stressful stimulus, compared to the parasympathetic nervous system, which is kind of the opposite. When parasympathetic nervous system takes you back towards homeostasis, it takes you back towards the normal baseline levels. The parasympathetic system is when you're no longer stressed and you're returning back to the baseline levels of not being stressed. That's the two divisions of the autonomic nervous system: sympathetic, which is fight or flight, and parasympathetic, which is homeostasis, returning back to homeostasis. So those are the divisions of the nervous system. Okay, so here we're going to talk about the brain stem. And the brain stem is located at the very bottom of the brain. Like the name implies, it is kind of like the stem of the brain. The spinal cord, I'll show you the spinal cord. Here's the spinal cord again. The brain stem is basically right on top of the spinal cord, right there. So it's like the stem, or the lowest part of the brain right there, connects to the spinal cord. And that's what this mostly this stuff is right here. This whole thing is basically considered the brain stem. That's the bottom of the spinal cord right there. And then the brain stem sits right on top of the spinal cord, which ended right there. And the cranial nerves have the brainstem has several components that we'll be interested in. One, these cranial nerves. These cranial nerves are, let's see, that's an example of one right there. Their nerves kind of start at the brain stem. Some of those nerves will go up into your face, and they'll do things like allow you to move your mouth. They'll also allow you to feel sensation on your face. Cranial nerves are also important for, like, when you taste something or smell something, those messages are taken from the receptors on your nose or mouth, and then they're passed into your brain via these cranial nerves. Actually, I want to show you, I think I have pictures of those, and I'll come back to that. So yeah, here's an underside picture of the brain, and you can see some of these cranial nerves. So this whole thing right here, this is the brain stem. So the spinal cord would be, this is like the spinal cord is removed right there, and then all this stuff is the brain stem. These branches that are coming off of the brain stem are the cranial nerves. A lot of these cranial nerves go into the face, but some of the cranial nerves will also go down, some of them will go down into the body a little bit. And that's what it says, cranial nerves, connections to various parts of the body, especially the face. The medulla is another part of the brain stem, and it's important for keeping you alive. If you have a stroke in the medulla, that's like the one area of the brain where you're most likely to die if you have a stroke, because the medulla basically keeps your heart beating. We don't normally think of the brain as keeping us alive, but we usually think of the brain, and the brain is important for thinking and planning and moving. But the heartbeat, which is a type of movement, is there's a pretty specific region of the brain. Once again, this medulla. Let's see, we have right there, which keeps your heart beating. The midbrain is another part of the brainstem. I don't have too much about the function of this, but that would be above here. So the midbrain is up a little bit higher compared to the medulla. The medulla is low, and then above that is the brainstem, and then above that is the thalamus, which technically is slightly above the brainstem. And the thalamus, we will definitely talk more about the function of the thalamus in later chapters. To kind of give you a sneak preview, the thalamus is kind of thought of as like a relay station. Many signals will start in the thalamus and then be transmitted to various parts of the brain, and those various parts of the brain will oftentimes send signals back to the thalamus. So it sends signals out and back in via the thalamus. So we'll definitely learn more about that later on. So these are all, some of the lowest parts of the brain, physically. Once again, all I was just talking about is all about right here. Here's another picture of cranial nerves, by the way, especially the ones that are going to the face. But you can also see the vagus nerve, which goes down to the heart down here. So another brain region that we'll talk about a lot, especially in the chapter on motor behavior, is the cerebellum. And all I'll say about it for now, we'll study the role of the cerebellum later on the formation of long-term procedural, that is motor memory. So like your ability to do things like learn how to play the motions that are required to play a musical instrument, or the motions that are required to hit a golf ball or to serve a tennis ball or to do any type of musical or sporting behavior, or a dance, any type of motor behavior which is well learned and requires a lot of practice. Your cerebellum is an important region of the brain for acquiring that motor skill. Cerebellum is also important for balance, any type of coordinated motor behavior, like I just said, which is kind of synonymous with procedural memory, especially memories of motor behaviors that take a lot of practice. The cerebellum seems to be involved with acquiring that skill, that motor skill. Moving on, and the cerebellum, you can see it. The cerebellum is this red area right here. That is the cerebellum. It's very close to the brainstem. Brainstem is slightly below and slightly in front of the cerebellum, and the cerebellum is slightly above and behind and then back of the brainstem. So the brainstem and the cerebellum are like really, really close to each other in the brain. Moving up, so that's kind of the organization here. We're starting at the lowest regions of the brain, which in theory, those were the regions of the brain that evolved earliest. And then we're moving up towards, here we're in the limbic system. Eventually we'll be moving up towards the frontal lobe, which is like the highest part of the brain, which is the region of the brain which is most developed in humans, the frontal lobe. But for here, we're talking about the limbic system, which is, now we're getting into what's known as, we're getting closer to the cortex, those outer regions, the top part of the brain. But here we're still kind of in the middle of the brain. So this is a good cross section of things right here. You can see the thalamus, and I mentioned earlier, the thalamus kind of sits on top of the brainstem. So the spinal cord would travel up through here. The brainstem you can't actually see because it's in the middle of the brain, but the brainstem would be about right here in the middle of the brain. On top of the brainstem is the thalamus, still moving up and then towards the sides. And the brainstem is right in the middle, moving up and then towards the side. We're moving out towards the outer parts of the brain. Is the limbic system, which contains these regions of the brain. We will definitely study the hippocampus. We'll study in a lot of detail because the hippocampus, there's some really interesting research. You might have heard of HM, patient HM, who had his hippocampus damaged and then had some very severe problems with encoding or learning new long-term memories. The amygdala, so that's the hippocampus right there, is part of the temporal lobe. The amygdala, which is adjacent to the hippocampus, but it's anterior, that is, it's in front of the hippocampus. The amygdala is really important for the perception of emotion, particularly fear. Individuals with damage to the hippocampus, to the amygdala rather, will see something which is most people would have a strong fear response to, but if your amygdala is damaged, you'll be like, meh, it won't be that scary to you. And then the cingulate gyrus, another region right here, which is particularly important for kind of making you pay attention to stuff that previously you've learned is important, or things that you've learned that previously that stimulus is rewarding. It does things that I like. The cingulate gyrus kind of directs your attention towards things that you think are important. We'll go into more detail about all of these later on. Once again, this is just kind of a rough sketch of the entire brain. So here is the cerebral cortex. The cerebral cortex is typically the name given to the outermost layer of the brain. So we talked about the brainstem, which is down here. We've talked about the thalamus, which is above the brainstem. We talked about the limbic system, which is, slightly more towards the outside of the brain relative to the thalamus and slightly above the thalamus. We're moving, up and out, and eventually we're getting towards the cerebral cortex, which is just this outer layer of the brain. And there's a lot of these anatomical areas that are used to kind of partition the cerebral cortex into different segments. The one that I care about, knowing the most is the precentral gyrus and the postcentral gyrus and the central sulcus. So gyrus basically means like it's like the bumps right here, versus a sulcus, which is like it's space. So the sulcus is the space between two gyri. The gyri is the actual, folds right here. And then the sulcus is the area in between them. And so usually in the past, for instance, I've had a question on test 1 that requires students to label these areas right here: precentral gyrus, the central sulcus, and the postcentral gyrus. And the reason I care about the precentral and postcentral gyrus is because there are two regions of the brain which are housed right there. Here's another view, by the way. The central sulcus right here. This is like this view right here. This is obviously a side view of the brain. This is a top view. So if you're looking down on top of someone's brain, you can see this, the interhemispheric fissure, which separates the left and right hemispheres of the brain, also called longitudinal fissure. But you can see the central sulcus right here. It's basically the crack in between the precentral sulcus, which is right here, and the postcentral sulcus, which is right here. And the regions of the brain that on the precentral sulcus, the precentral gyrus, I'll go back to this one because they're talking about the sulcus is the crack right there. The precentral gyrus right here, this is where an area of the brain called motor cortex is housed, motor cortex. And the precentral gyrus right here is where somatosensory cortex is. So there are these two regions of the brain. The motor cortex is involved with voluntary movements, being able to move your limbs, like when you want to. And the somatosensory cortex, which is right here, is important for those different limbs to be able to feel, touch. So this is the region of the brain that allows you to move different areas of the body. This is the part of the brain that allows those different parts of the body to feel things. They're separated by this gap, by this crack, the central sulcus. So these are the areas anatomically that I'll care most about. And we'll talk a lot more about the motor cortex in particular in the chapter later on action, on motor behavior. Questions or comments about what I've said about the cerebral cortex here?
To clarify, you said pre-central was where the motor cortex was and post-central was for the limbs to feel?
Yeah, somatosensory is where the limbs feel, and that's post-central, yes. And I'm going to show another, there's another diagram right here, which completely disambiguates anything I just said. Motor cortex right here, here's pre-central gyrus. Then the central sulcus is the crack in between them. And then the somatosensory cortex is in the post right here, the post-central gyrus right here. A motor cortex, as the name implies, this is the part of your brain that allows different body parts to move voluntarily. Like right now, I'm using my, motor cortex to do that. But when I feel something on that same region of the brain or same region of my hand, it's the somatosensory cortex, presumably, which is important for representing the feeling of touch. And then over here, the last thing I guess I'll say about this, the frontal cortex is all of this stuff up here. Our frontal cortices in humans are quite big, bigger than basically any other animal. And so it's definitely, a lot of people believe, including myself, that a lot of the things that separate us from basically every other animal as far as like language and the appreciation of music, the ability to plan for the future, those are things which the frontal lobe seems to be really important doing. A lot of the things which make us uniquely human appear to come from the fact that we have particularly large frontal cortices. And especially the very, very, very most frontal part of the frontal cortex is called prefrontal, which basically means like before the front. Those are particularly pronounced in humans. We have big prefrontal cortex. For now, I don't care about these subdivisions of the frontal cortex other than motor cortex and somatosensory cortex and the central sulcus. So yeah, a little bit more about the motor cortex and the somatosensory cortex then. The Montreal procedure, which is something we learned about in chapter 1, that is a procedure, Penfield, I believe was the name of the individual that found it, which you can electrically stimulate different parts of the brain. And the Montreal procedure was originally used to kind of help diagnose what part of the brain is causing a seizure. But when this Montreal procedure was used on motor cortex and somatosensory cortex in awake people, so they actually had individuals that were awake, but they had strong local anesthetic on, and their skulls were opened up, so they weren't in pain. These people would have, while they were awake, they would have the motor cortex and or the somatosensory cortex stimulated by the Montreal procedure. And what the researchers realized, medical doctors and researchers realized, was that they could elicit certain behaviors as a result of stimulating these different regions of the brain. And here is a kind of an illustration of what happens when the motor cortex is stimulated. This is like across a coronal slice of the motor cortex, and you could stimulate these different regions of the motor cortex in the Montreal procedure. And if you stimulate this area right here, it'll cause your arm to move. If you stimulate this area right here, it will cause your wrist to move. This area, your hand, this area, your fingers, this area, your neck, this area, different parts of your face, this area, your tongue. If you do this area, presumably it can cause you to swallow. So all of those different voluntary motor behaviors that we have, each one is kind of housed in a different area of the motor cortex. And using the Montreal procedure, which is electrical stimulation of these different brain regions, you can actually cause a given person who's awake when they're stimulated to move each of these different parts of their body. So this is what a topographic correspondence is, when basically the organization of the body is preserved in the organization of the brain. A similar thing happened in somatosensory cortex using the Montreal procedure, except when you stimulated these parts of the somatosensory cortex, it didn't cause the person to move, but it would cause them to have phantom sensations. So you could actually stimulate these different parts of the brain. It would cause the person to feel like they were being touched on their head. Or if you stimulate this part, you feel like they're being touched on their hand, or they're being touched on their, they're getting a sensation in their eye, or in their mouth, teeth. So you can feel, or their tongue, depending on where the stimulation is occurring, you can basically cause the person to believe that they're getting some type of tactile stimulation somewhere on their body, even though they're not, which has led to the kind of characterization. This part right here, this is the motor cortex, voluntary movements, versus the somatosensory cortex, which is involved with feeling, sensation. A couple more things, then we'll get out of here. Visual cortex, this is another region of the brain. This is in the occipital lobe right here. So this is the back of the brain, like right in the very back. You can't see it, but the cerebellum would be right here. The brainstem would be about right here. Cerebellum right there, brainstem here. And then the occipital cortex is basically this. And in particular, right around here, kind of in the middle part of the occipital lobe is primary visual cortex. We'll learn a lot more about primary visual cortex in, I believe, chapters 5 and 6, also known as V1 or striate cortex area 17. It's basically I don't want to say it's the very first place that visual information goes, but it's one of the very first areas of the brain that visual information from your eyes goes to. So it goes from your eyes, and then it actually goes to the brainstem first and the thalamus. But then the first kind of cortical area, you know, the outer cortical area, the very first cortical area that visual information goes after it enters your eyes is your occipital lobe, which is where primary visual cortex is, about right here. Analogously, here's the auditory cortex, the primary auditory cortex. So the visual cortex is over here, occipital lobe. Auditory cortex is right here. This is the temporal lobe right here. And it's kind of analogous to visual cortex in the sense that it's basically the first cortical region where auditory information goes. So auditory information goes from your ears, and then it goes to your brainstem, and then eventually it goes to the outer cortical region, that is the temporal lobe, particularly the primary auditory cortex, which is right here. So these two regions, visual cortex, auditory cortex, we'll talk in a lot more detail about what type of visual and auditory information they represent later on. But that's it for today. We finished chapter 2. I know it was a lot of lecturing. I had to catch up. We were just slightly behind. We're basically caught up now. So we will be starting chapter 3 next time. And there is, as usual, quizzes to take a look at. Oops, that's wrong class. I already shut out the syllabus. Let's see if I can get it back. Yep, textbook chapter 3, I'll remind you, that is due Thursday. I've extended the deadline for that. Instead of it being due Wednesday night, I've made it due Thursday morning at 9:15 a.m. So that one is due Thursday morning.